Welcome to the Jack Westin MCAT podcast with your host, Phil Hawkins.
And Azaii Muniz. Welcome to the next biology-related episode of the Jack Westin MCAT podcast. We've been really excited doing this mini-series inside the podcast and getting to walk through some of the core topics in the biology, biochem section, things that can come up in other sections and have those little tie-ins, but really talking and getting down to the central dogma. And we've talked about how we go from DNA, which are the instructions in our cells, to the RNA, specifically the messenger RNA. And those are taking specific instructions. So how to build a chair has been the prevailing example. And to how we actually build the chair, how we build the proteins that make up our body that ultimately make us. And we've talked about these steps individually, but today we're going to take a look and take a step back and think about how do we control which proteins are made, how many proteins are made, what these proteins do. And this is something that's incredibly important because, like we've chatted about, each cell in our body contains the same complete set of instructions, but within each cell, some of those instructions are carried out, others aren't. And at different points in our lives, we might have instructions carried out more frequently, sometimes less often. And it's helpful to be able to understand these things, yes, for the MCAT, but also for thinking about your future career, for future research you might want to do. And there's a lot here that we're still learning, but a lot that we can learn together.
Yeah. This is a big deal, and the MCAT really likes to test this, and it is very easy to get twisted around with these things. And so the important thing is to realize that every step that we've talked about over the past couple of episodes, like we can control that step specifically. Like you can mess with transcription, right, where I'm making more messenger RNA or less or none even, where you can kind of change what's going on there depending on how much of a protein you need. Yes, all the cells in your body have the same DNA, but different ones will have different amounts of protein. And like you might have one cell that has a lot of a protein, so we need lots of messenger RNA. We might have another cell that's got some of that protein, but not quite as much. And so we need a little bit less messenger RNA. And some cell might not be making that protein at all. Now, what's interesting is most of the time when we think about regulating proteins, we think about regulating transcription. Just making messenger RNA or not, right? So we can have this messenger RNA, we can make those photocopies of our instruction set at the library of the nucleus and like look through all of this stuff. Like, yeah, we want this. And so we'll make a copy of that. That's our messenger RNA. But what's interesting about this is not only does this stuff happen in different cells in your body, but there's a lot of things that can change in your body over time. And even be carried between generations. And this is a super interesting topic called epigenetics, where like if I were to do certain things that is going to change the activity of my genes, right? Like if I'm exercising a whole lot, I may adjust and change what levels of different proteins are in my body. Like if I need more energy for running, I may increase the amount of the enzymes responsible for producing energy, like glycolysis and Krebs electron transport chain. And even just like metabolism. If I haven't eaten in a while, I can change that. But what's interesting is you can sometimes pass these genes down to your kids. And so there's some inherited things. And so what's interesting is that the genes themselves, what genes I have, I'm always going to pass down my DNA to my kids. But I can pass down more active versions or less active versions, which means that what's going on in the parent's life could affect what's going on in the child's life afterwards, like what's going on at the level of DNA transcription.
Yeah, and this is something that when you're having conversations with patients may not be the first thing that comes up. But especially if you are someone that is going to be working in obstetrics and gynecology and working with pregnant patients, having a conversation about, hey, the things that you're doing can affect, right, the growing fetus and your future child. And this is one of the ways that you don't have to go into super deep dive with your patients, but even just letting them know, yes, let's work together to minimize the level of stress that you're experiencing. So we know, and we've talked about this on the podcast, that having that constant stress response can have negative impacts on our health. And so thinking about how that can be transferred, right? Thinking about how blood glucose levels, right? The amount of sugar in the bloodstream and the amount of insulin that has to be produced and how the cells respond to all of that extra insulin can cause changes in how the fetus develops. And we know this, right? And so sometimes just having that very brief conversation with patients can make a really big difference, because a lot of times we have all of this extra knowledge and this understanding of how and why that our patients don't come in with, because they didn't study to become doctors more often than not, right? And so being able to communicate that can have a really big impact. So don't consider these topics in isolation. They are helpful.
Yeah. I love that you talked about the glucose stuff. That's actually one of the most, one of the most applicable versions of this. Like, what's going on with the mother while she's pregnant will affect the fetus, and not just while the fetus is in utero, but also outside of that. It turns out that the mother's diet while pregnant can increase or decrease the odds of the child getting diabetes later in life, right? 10, 20 years down the line, based on what's going on with the mother. And so that's kind of an interesting thing, because our body is constantly adapting to the environment around us, and we are changing we're not changing our DNA. It's always the same sequence, but we are changing our expression of that DNA based on these environments that we place ourselves in, or in your fetus case, in the environment you are placed in, because they don't have a lot of control.
So then the question kind of becomes how, right? Because, okay, yes, these changes can be passed down. We have to think about how one of the ways at the level of transcription that our body can say, okay, yes, you can transcribe this gene more often, or no, hold off, I don't want you transcribing that gene as much. I like to personify the sciences, so obviously it's not a perfect example. But as you probably remember, our DNA is not just sitting around, unwound, kind of hanging out, fully unfolded in the nucleus. There's a lot of DNA and not a whole lot of space. And so one of the ways that our body handles that and allows for this control at the level of transcription is by winding our DNA and our chromatin around what are called histones. And histones are these basic proteins that so they're positive, right? That can interact.
Basic in more than one way. Basic as in alkaline, and also basic as in simple.
Yes, both. So they have lysine, a basic amino acid. Know your amino acids on test day. We say this. You're probably tired of hearing us say this, but we can't stress it enough. And so they're going to interact with the negatively charged DNA. And these histones, you put a couple of them together, you get nucleosomes, and the DNA can bind around these, and they form these points where, when the histones are modified, they can either let go a little bit or hold that DNA tighter. And so this is done through methylation and acetylation of the histones. And so methylation is going to cause that chromatin to bind tighter. Acetylation is going to cause the DNA to loosen up a bit. And so if the DNA is bound tighter, then it's not going to be transcribed as often. Whereas if it's looser, then it will be able to be transcribed more often. It's going to be more prone to it. So you have these two different modifications that can happen at the level of the histones, the methylation and the acetylation. You have to recognize that the histones can be methylated or demethylated, acetylated or deacetylated. And so when you remove the methyl group, you're going to have the opposite happen. So instead of binding more tightly, it's going to loosen up a bit. If you remove the acetyl group, then it's going to tighten up a bit instead of being released as though it's it compared to when it were acetylated. And so this is a way that at the level of the histones, at the level of how tightly the DNA is bound to these little points, that can impact how often something or a gene is or isn't transcribed.
Yeah. There's a lot of crossover here into other topics.
Yes.
Right? Like, obviously, amino acids are big in this. Like, the MCAT likes to give passages about methylation or acetylation of histones specifically, and they're expecting you to have some good working knowledge of that. But it's also really easy to take this into the realm of orgo as well.
Yeah.
So note that lysine has this like long chain with a nitrogen on the end. So it's got an amine group. And that amine will just pick up hydrogens, like often happens with amines. Like, that's kind of how they tend to default to either neutral or just positively charged because they've got an extra hydrogen on them. But whenever you acetylate a histone, you're turning that amine into an amide. And that's going to create this interesting resonance structure where that nitrogen doesn't want to pick up a hydrogen because it wants to be able to donate those electrons to that bond to the carbonyl. And so that stabilizes this and makes it not want to pick up a hydrogen. So it loses that positive charge. And so, obviously also that connects to electrostatics with like positives and negatives. The DNA is negative, the histones are positive. If you acetylate them, they lose the positive. So they just fall apart because it was the charges that were kind of holding everything together. And so those are big deals, right? Being able to kind of understand the connections to it, because they can definitely give you this passage about a histone and then start asking you questions about electrostatics or questions about organic chemistry or questions about amino acids. Honestly, they're probably going to ask you an amino acid question. They might also have one of the other.
They should.
Yeah, they should. Anytime there's anything that can be connected to amino acids, they pretty much always connect it to that. But it is interesting to note that it isn't just modifications to histones that we can change. It's also modifications to the DNA itself. One of the ways that we can modify DNA is by methylating it. So we just add a carbon to it, add a methyl group to one of the bases. What's interesting is this, like modifying the DNA, whenever you add a methyl group to it, it makes it harder for RNA polymerase to read the DNA, to make the messenger RNA. Because it's like going along looking for A's, T's, C's, and G's, and it runs into some weird thing with a methyl group on it, and it's like, I don't know what to do with this. And so it ends up not working as well. So methylating DNA is also a way that you can modify the rate of transcription of a protein. I will say this methylation one is actually the one that is more responsible for the passing down of modifications to genes. The histones are proteins, and so those don't tend to stick around as long. But if you methylate DNA, like that DNA is going to get passed down to your offspring, right? Because of how the whole system works. Is like half the DNA comes from mom, half the DNA from dad. Some of those genes are methylated. Those are going to get passed down as well. And so it's kind of an interesting concept to be able to kind of understand and connect to that. Side note, there are some... I'm like, we weren't planning on talking about this, but this is something that I have seen the AAMC test. There are some genes that we need just one copy of, right? A great example of that would be what's going on with the X chromosome in women. Like, they need just one copy of it, so they tend to just completely shut down one whole X chromosome. And so they'll wrap it up real tightly around histones and just kind of silence that entire chromosome by condensing it. Now, what's interesting is there are times, and that's a good example because like everyone needs one active X chromosome. Men only have one, so we're good. But women have two, and so they need to silence one of those. But note that there are genes that we need one copy of that aren't on the X chromosome. Genes on like chromosome 1 or 2 or 3 or 4. And so what happens is in the process of making a sperm or making an egg, one of them gets methylated, right? And so if I'm making a sperm, the human evolution has created a system where it's like, okay, we know we need one copy, so whatever the dad's passing down, silence it. And so they will just methylate that gene so that then when you get the offspring, they're just getting the active version from the mom. Now, that would be a maternal imprinted gene is the term for that. There's also paternal imprinted, where the dad's passing down the active and the mom's passing down the inactive. And note that this is not a normal Mendelian genetics scenario. This is non-Mendelian genetics is actually the category I would kind of like lump it into. And so it doesn't follow the normal Punnett square stuff, where it's something interesting where mom or one of the parents is passing down the gene, but it's inherently inactive. And so it only matters what the other parent is passing down to determine whether or not the person has the disease or not. So that's also something I've seen passages about, and this idea of imprinted genes is something that the MCAT has asked about, and it's kind of a strange thing that most genetics professors don't even talk about.
Yeah, I love that you mentioned that. As soon as I don't know if you saw my face, I was like, oh.
I did.
As soon as you started talking about that, I was waiting. I was like, okay, we have to talk about exon activation. And it wasn't part of our plan, but the formation of our bodies. And again, there is a way that you will use some of this information we just talked about imprinting in your future careers. Mm-hmm. Some of the illnesses and disorders that we see are a result of maternal or paternal imprinting, and knowing to ask about certain family members and, learning to ask about the paternal lineage or the maternal lineage when you sit down with a patient, something we don't often do explicitly, but that can help you pick up on, they might have this disorder. They might be more likely to have this disorder. And so this information, again, does translate to you being a future physician, even if very subtly.
Yeah. And so this is why it's important to have a good conception of this. So another way that we can regulate transcription, right? So all of this is talking about ways we can regulate making the messenger RNA. More of it, less of it. You can acetylate histones, you can methylate histones or methylate DNA, or you can kind of do things like that. But another way is transcription factors. So just there are other factors in our body, and this is how we respond to the environment around us, is they will come in and they will bind our DNA, and they will either increase or decrease the expression of certain genes. It's really interesting. If I am in starvation, I'm going to be breaking down a bunch of fat because my body, that's the best source of fuel if I'm in starvation. That's why we store fat, is in case we're starving later. And so that will actually, the products of that breakdown, the beta-hydroxybutyrate and acetate, these short-chain fatty acids, will start to interact with things in our body, and it will start to modify our DNA expression or transcription of DNA. And so in early starvation, you actually don't have that many proteins to break down fat. And that's because under normal scenarios, you don't need a lot of proteins to break down fat. But when you go into starvation, your body actually over time will start to build more and more and more of these proteins for breaking down fat. We don't want to do it all the time because it's kind of a lot of energy and it's wasteful to be building stuff we're never going to use. But when we're starving, we need a ton of these. And so our body will adjust to that. And they do this via transcription factors, where different things will come in and they will change expression. There's a lot of examples of transcription factors. Honestly, that's also a topic that is all over on the MCAT. Note that steroid hormones tend to work that way. So why I, developed as a male is due to testosterone, interacting with my DNA via some proteins, to adjust and change the transcription of certain genes. And likewise in women. Note that men and women both have the same genes for muscle, right? They're same myosin, the same actin thing, but men tend to have more muscle. That's because certain hormones will act as, will interact with transcription factors to increase the expression, increase the transcription of the messenger RNA responsible for building muscle. And so this is how we're able to kind of adjust to different scenarios.
Yeah, there's, like you mentioned, there are differences that the amount of a hormone produced can influence. You do need to know the different hormones for testing. You need to know the difference between steroid and peptide hormone. You need to know what testosterone looks like. You don't have to be able to draw it from memory, but it's a steroid hormone. You should recognize cholesterol somewhere in there. So there's a lot that we encourage you to take a look at outside of this podcast episode. Not the time for us to dive into it. But make sure that you are familiar with different hormones, not just steroid hormones, right? The ones that act at the kidney, the ones involved in calcium homeostasis, etc. Yeah,
we have talked about steroid versus peptide hormones in previous podcast episodes, so feel free to hunt down those episodes.
Yep. Also muscles and bones.
Yeah.
So we've talked about this next topic fairly recently in the recent series, so we're not going to spend too much time on it. But another way that we control transcription, specifically in the process of creating that mRNA is this idea of alternative splicing. So we said that mRNA, before it gets finalized, it has introns and exons. The exons are the coding portions. The introns are not coding. And we are able to remove some of those removing the introns, we're able to remove some pieces altogether. We are able to keep them in other variants. And so we can form different versions of the same protein. And this alternative splicing occurs before we actually start translating translating the mRNA into the protein. So this is another way that we are controlling which proteins, in this case, which version of proteins are being produced. If you're a little rusty on transcription, I think might have been the first episode that we did in this mini series, so you can take a look at that.
Yeah. So that is, there's something really interesting here, because I want to kind of touch on this. For a long time, we thought introns were trash. And we're like, why is most of our DNA this useless stuff that isn't actually a coding sequence? And it turns out that they are not trash. They are really useful in a lot of different ways. One of the ways, so if I'm making protein A, right, and as I am transcribing that, I am cutting out some introns from that. The introns from that protein A could combine with certain proteins. And those proteins are what we would call spliceosomes, so those spliceosomes are now going to be a mixture of protein and the RNA from the intron. And so this is what we would call a riboprotein, which is like RNA, like ribonucleic acid, and also protein. It's a mixture of the two. And so this spliceosome, which has a chunk of RNA in it now, that's not just pure protein, know that RNA, like if it's like U, that's going to bind to a strand of DNA that's AAAA, or a strand of messenger RNA that is AAAA. And so this intron that we cut out, we incorporate into the spliceosome, and now it's going to bind different spots on messenger RNA. And so it's going to change the way that we cut and splice messenger RNA. So it's really interesting that building protein A can affect the splicing of protein B, C, D, and E. And so by making a certain protein, we're adjusting what messenger RNAs we're making with the other genes in our body. And this is a super interesting, kind of crazy fact. And I remember when we first kind of discovered this, it was like, whoa, like this is something really interesting. Like all of a sudden, if I start making insulin in my body, I'm not just making more insulin, I'm also changing the other proteins that I am making by changing the transcription and the splicing in those other genes. And so all of these are like transcriptional controls, like where we are controlling transcription. We're affecting the splicing, we're affecting whether or not we're just making it or not with like transcription factors or histones, like being acetylated or methylating DNA. All of these are specifically transcriptional control, but this isn't the only way we can control whether or not we make a protein. So the other way that we control this is controlling translation, right? So just reminder, DNA to RNA or messenger RNA, that's transcription. Once we have that messenger RNA, turning that into protein is translation. So a way for us to control this is via something known as siRNA or miRNA, microRNA. And so what happens here? So this is also, interestingly, based on introns. Let's talk about microRNA first. So if I am making protein A, I can cut out the introns from that. And those introns might bind other messenger RNA, not with the spliceosome attached, but just generally, right? So if one of my introns is, let's say, CCCC, if I have another messenger RNA that has GGGG on it, then that will bind and I will make a double strand of messenger RNA. You can make double strands of messenger RNA. I just want to reiterate that. I know a lot of times students are taught or told that like DNA is double strand, RNA is single strand, like most of the time, but like you can make double stranded RNA and single strand DNA can exist, just not very often in humans. But looking at this, now once we have a double strand of RNA, it's the messenger RNA and this thing that we will call the microRNA, which is this intron from another gene. that can bind, and now we have a double strand, and now a ribosome can't read it because ribosomes read single strands. And so what we've done here is, even though we're making this messenger RNA, we can't translate it. So we've stopped translation. I know a lot of times this seems like a lot of pain and kind of annoying. Like, why don't we just stop making the protein? But this allows us to have some adjustment, right? So like, let's say this isn't exactly how this works, but this is something that could work, and it just should help you kind of conceptually understand this. So let's say that I haven't eaten in a while, and I'm making a bunch of glucagon, right? Because I need to raise my blood sugar. And so I'm in like this starvation phase, and I need to raise my blood sugar because I need to make sure that my neurons have glucose, because if they don't, they die and they don't come back, and that's kind of a problem. So what I need to do is make a bunch of this glucagon. So if I eat a bunch of donuts after that, right, what's going to happen is my blood sugar is going to spike, and I don't need to keep raising my glucose. So I need to start making insulin instead. And so when I make the insulin, I can make that messenger RNA. I can cut the introns out of that. Those introns might interact with the glucagon messenger RNA. And so even though that glucagon messenger RNA is still out there because I was just making a bunch of it, when I start to make insulin, that's going to create some introns which might bind the glucagon messenger RNA. And so even though that messenger RNA is still out there, I stop making the protein. So I stop making glucagon because I started to make insulin. And so this is just a really advanced way for us to control the levels of proteins and do like fine-tune adjustments very quickly. Note that I think about microRNA is they're not super specific. I mean, they are specific, but often one microRNA might bind like 3 or 4 different types of messenger RNA. And even if I'm making that microRNA, I'm still going to be making like messenger RNAs. Some of those messenger RNAs will run into microRNAs. So it's not going to completely stop me from making the protein, but it will make me have less of the protein. There is also like a synthetic version of this, something that we will use in labs that is known as siRNA. I always remember that the S stands for synthetic. It doesn't actually. Si stands for small inhibitory RNA. But this is kind of the same thing as microRNA. MicroRNA is naturally occurring. It's made from the introns that we cut out of our messenger RNA as we splice them. siRNA is something you can buy from like a lab, and they will send it to you. And usually they are really specific to just one like messenger RNA. So I could buy siRNA that will bind like the insulin messenger RNA. And if I inject that into a pig, now all of a sudden this pig is going to be making way less insulin because the siRNA is going to be binding that messenger RNA. So essentially what I've done is I've created a pig that is a type 1 diabetic because all of a sudden they're making way less insulin. Like we have this kind of drop in insulin. This is something called a knockdown, which you do want to be familiar with, which is different than a knockout, right? A knockout is when you remove the gene from an organism. Like, hey, it's gone. They're out. They have none of that thing. Versus a knockdown is when you use siRNA, and that greatly reduces the amount of messenger RNA, or that is active, greatly reduces the amount of protein. And so you've knocked it down instead of knocked it out completely. And so those are like ways that we can control specifically translation by adjusting whether or not we're making this protein. Even if we have the messenger RNA, we can still stop ourselves from making the protein using microRNA, or if we're in a lab scenario, siRNA.
And this is also a great place, to your point, Phil, with being synthetic in a lab, knockdowns, knockouts, for them to throw in some data interpretation in whatever passage mentions si or microRNA. And so if that's something, this is something that I've seen a number of students have difficulty with in terms of data interpretation, figuring out how to follow the graphs that show different either amounts of proteins or protein activity following knockdowns, following knockouts, heterozygosity. So if that's something that you're listening to this and you're thinking, ooh, I might need to brush up on that, make a note of it, and do take a look at it, because it's a place where you could gain easy points or lose easy points.
Yeah, it's something I've seen the MCAT test quite a bit with messages about microRNAs. Maybe they just give you a bunch of sequences for microRNAs. There is a whole lot of connections between microRNAs and different diseases and expressions. Even things like whether or not you're likely to reject a transplant, like what microRNAs are in your body, that's going to adjust and change the proteins and your immune system. And so it's one of those areas that's kind of like almost cutting edge, where we have learned to kind of look for different amounts of proteins and relate that to different cancers and different things as well. But some of the ways that these things work might not be via just protein overall. It might be we are adjusting translation, and it might be a lot more about microRNAs instead. And so there's a lot of research on this, and because there's a lot of research on it, that means it tends to show up on the MCAT, because the MCAT doesn't pay people to build their own journal articles. They use real research articles. So whatever stuff is being researched a lot tends to show up more on the exam.
Absolutely. So we've chatted about how our bodies control protein production, amounts, whether it's produced at all, at the level of transcription, at the level of translation. But this is also the control continues after the protein has been translated. And so there are different modifications that can be made to this chain of amino acids, right? This protein that will influence where it goes, that will influence whether it gets to stick around or not, or is degraded, that will influence whether it is active or inactive. And so some of these are going to be lipidation and glycosylation. These can help the protein kind of direct them. So if you think about adding lipids, right, these are going to be, you should be thinking of the cell membrane as a potential target. They can help anchor it in the cell membrane. If you think of glycosylation, these are often cell markers. And so thinking about our blood types, we did an episode on that as well. You can also have phosphorylation. And so adding a phosphate group, depending on the protein, can activate it or inactivate it. This is something that you'll see, in other components of signaling, perhaps something that we are going to be talking about later. And we can also mark proteins for destruction via ubiquitination. There is, we can also, in addition to these modifications, we can cut proteins, right? We can, this proteolysis can cause activation, inactivation. And there's one example that I think is really important to know, and that's insulin, the production of insulin. So insulin is not created as the active insulin protein. Similar to how when we talked about the digestive system, a lot of those enzymes are not created active. In the case of the enzymes, we don't want them digesting us. We want them digesting our food. But we also don't want a bunch of free-floating insulin all of the time, right? We do need to produce some basal level insulin, but we need to control how much of it is active. And so it's produced in this pre-proinsulin form. And we have this first set of cleavage. We produce proinsulin and then cleave again, and we produce insulin. And one of the byproducts of this cleavage is C-peptide. So this was one of the most interesting cases that we did in, I think it was my first year of med school in class. They were trying to get this idea of C-peptide and insulin and how it ties into what you might see in the clinic or in the hospital, perhaps in a hospital setting with someone with low blood sugar. So there are a couple different scenarios that can happen. Someone might have accidentally ingested or injected too much insulin, right? They might have an insulin-producing tumor. They might have taken too much of a medication that causes insulin to be released. So thinking about someone with diabetes versus someone without diabetes. And using the amount of C-peptide, you can actually start figuring out, is it that they have an insulin-producing tumor? Is it that they injected too much insulin? Why? Because if your body is the one producing the insulin, there's going to be a ton of this C-peptide left over. But if someone accidentally injected too much insulin, they already injected the active insulin, right? So they're not going to have that C-peptide, all of that C-peptide floating around. I think in the example they used, it ended up being either someone trying to poison the person just to make it interesting, or the person took too much of their medication. So in that example, there was not a lot of C-peptide floating around, but you can find a scenario where you're asking yourself, okay, this person, as far as I know, doesn't have diabetes, but they're having episodes of low blood glucose, right? And that's something that we might see with someone who is taking medication. But in this case, they might just have an insulin-producing tumor in their pancreatic beta cells, right? So there are ties in that because you know how the body produces insulin, because you know that there's this modification and this activation process that needs to happen, you know what other tests to order, right?
Yeah.
And so there's a lot here. It's something that you can get on test day, and that could very well come up with your patients in the future.
Yeah. I've seen MCAT passages about C-peptides specifically because of some of the reasons that you're talking about. But that's also just something interesting to note. Like if you have a patient who like seems like they have a lot of insulin in them, but you know that like, oh, I know that their spouse is diabetic. Like that means like maybe there was a drug mix up and somebody injected themselves with insulin. They were supposed to inject themselves with something else. Or maybe there was, like you mentioned, like maybe it's a poisoning scenario. And like, obviously these are not like things that you should be immediately thinking all the time as a regular person, but as a physician, you need to be kind of aware of like, okay, like how can I eliminate options and figure out exactly what's going on here? Yeah, there are tons of ways to activate and inactivate proteins once you have them, right? You mentioned like phosphorylation is a big one. You can also acetylate and deacetylate. We talked about that with histones. Note that whenever you acetylate a histone, it makes it, I don't want to say deactivate, it just makes it unwind from our DNA because a histone isn't exactly an enzyme, so it's not really like an activity thing. But this is something that is all over the place. And so we may be making a whole bunch of insulin. We are transcribing lots of messenger RNA. We are translating that into like the protein, but we still might not have a lot of active insulin in our body because we haven't done the modifications that we need to. This is something that also comes up in a lot of times we need proteins for treatments. For example, insulin is a protein. And so I may use bacteria and like inject them with DNA that can make insulin. And so the bacteria is going to start to make insulin. So now I can take that insulin and give it to diabetics. And that works really well. But there are other times you might try to do this with other proteins. I'm like, oh, I got this from a bacteria. And then I put it into a human and it doesn't work. And it's probably because we need to do some modifications to that protein to actually turn it on. And the bacteria isn't doing those modifications. And so like it works in humans if they make it. It doesn't work in bacteria if they make it because it's not doing those last modifications. It's also worth noting that I know we've kind of hinted at this before, but there is a big difference between steroid hormones and peptide hormones in terms of how quickly they work and like how long they last and things like that. So with steroid hormones, they tend to be affecting transcription. So if I want to increase a certain amount of a protein and I'm doing it via steroid hormones, the steroid will kind of cause this transcription factor to activate and make more of that messenger RNA. And then we're going to take that messenger RNA and then we're going to translate that into protein. And then we're going to take that protein and we're going to do all the modifications that we need to in order to make that active protein. And so that takes a while to do all of those things. Versus peptide hormones, they come in and bind a receptor on the cell membrane, and that causes them to activate proteins that are already there. Cause you to like, oh, let's have this phosphorylation cascade. And everything gets phosphorylated, everything's turned on. All those proteins are already there. They're just waiting to be turned on. We don't have to wait for them to be transcribed and translated. They're all just sitting there, just waiting, just like, give me a phosphate and I'll go to work. Just help me out. And so when this peptide hormone comes in, it causes this cascade where they all get activated and they're all just sitting there waiting. And so that's why peptide hormones tend to work much faster in biology, is because they are creating active proteins or inactivating proteins by modifying that last step, that control of protein activity, versus steroid hormones are affecting transcription, which is way earlier in the process. And so that takes a while to kind of go through this. I do want to kind of mention one other thing here, because the MCAT really likes to ask about this. Often they will ask you about like, how do we measure transcriptional regulation, or how do we measure translational regulation, right? If I'm not making a protein, what part of this am I kind of stopping from working? Am I not making the messenger RNA, or am I making the messenger RNA but not making the protein? I'm not translating. So whenever you're doing these things, if I want to see if I'm controlling transcription, you have to know, do I have the DNA and am I making the messenger RNA? Now, the thing is, every cell in your body should all have the same DNA. So you kind of don't need to check, like, do you have the DNA for this thing? Because the answer is yes, unless there's some weird mutation going on. And so you only really need to check for the presence of the messenger RNA. If we aren't making the messenger RNA, and we don't have it, then we have a transcriptional repression where we are not transcribing. If we do have the messenger RNA, that means, we are making that messenger RNA. We're not repressing transcription. We're getting a lot of transcription. Now, if you want to measure translation, you can't just measure protein, right? If I measure something and there's no protein, like that doesn't mean that I'm stopping translation. I need to know, do I have the messenger RNA? And so you have to measure translation, you have to measure messenger RNA and protein levels. Because if you have the messenger RNA and you don't have protein, that means you are repressing translation, probably through a microRNA. If you don't have messenger RNA and you don't have protein, then you're repressing transcription, and you're not making that. And so there are lots of ways to measure these things, like blots and like RT-PCR and things like that. And I know that we've talked about those in other episodes, but I think it is important to kind of understand, like, because the MCAT asks often, like, how would you know if this is being transcriptionally regulated or translationally regulated? And so that's something that you want to kind of like check on and look for. Or in the case of insulin, maybe it's a post-translational regulation where it's like, we have the protein, but we don't have a lot of active insulin. Maybe there's something that needs to happen after that we need to modify.
Yeah. I want to leave you guys with one last mnemonic because, Phil, you reminded me of blots. And so there's no way I can't, share this.
Not mention it.
Yeah, I can't not mention this mnemonic. So I like using the mnemonic snowdrop to help me remember which blot corresponds to what we are looking for. So if you stack the word snow over the word drop, the letters directly correspond. The O's don't stand for anything. So be it.
Turns out you just need vowels for words.
Yeah, exactly. So the southern blot, or the S, corresponds to DNA, the D in drop. The S in snow, the northern blot, corresponds to RNA. And then ignore the O's. The W, so the western blot, corresponds to the P, protein. And so that's a way that you can remember what each blot corresponds to, just so you don't get anything mixed up on test day, because regardless, it's going to look like a black line. So you want to know what that black line on the blot represents.
Right. Yeah. So that's southern, northern, western. Snow is DNA, RNA, protein in respective order. That is one of our, I know we did a podcast episode where we talked about our favorite mnemonics, and that was definitely on our list. That's one of those like really high yield ones. And honestly, it's high yield because of these questions asking about regulating transcription and translation. So if you wanted to see if you were regulating transcription, you would want to check for RNA. So you would want to do a northern blot. If you want to check for translational control, you need to check for protein and RNA. So you need to do both a northern and a western. Now, there's other things, like you could do RT-PCR as another way to measure RNA. So there's other ways to check that, but still that same idea exists. Like you need to measure just RNA to measure transcription, but RNA and protein to measure translation. So I think that wraps up. Honestly, this was a kind of this like little mini series here where we talked about transcription in depth, translation in depth, how like what's going on in eukaryotes is different from prokaryotes. And now this last thing about transcriptional and translational control. If you guys have other thoughts for other like mini series that you would like to hear more about, please reach out to us at podcast@jackwestin, at jackwestin.com. Yeah, it turns out there's more to that. But we're excited to move on to a couple of other kind of mini series coming up. Hopefully you guys are excited to learn about that. We will be doing an episode very shortly on the updates from the AAMC and the announcements from the AAMC. I also want to point out, if you guys want to hang out with us in like the evenings, like throughout the week, we also do free sessions at jackwestin.com. You can always hop in, it's always great to like hear students who are like, oh, I'm coming from a podcast. I'm like, yeah, all right. And so that is always fun as well. We do it, generally, like Azaii and I do at least one a week, where we are kind of like reaching out. But
and you can find those on our homepage at jackwestin.com. You will also see sessions with other folks in here at Jack Westin, which can be really fun. And then, as always, you can find us doing some office hours in the course, and we are excited to see you then.